In this video, we're going to be learning about the structure and functions of the different parts of a neuron, which is just the scientific word for a cell in the nervous system. Now, neurons have probably the most easily identifiable structure, as there isn't really any cell that looks anything remotely like them. But as a consequence, even though this is actually a single cell, sort of, each of the different parts is relatively specialized towards the idea of transferring action potential from one part of the body to the next, and therefore we need to talk about the different parts of a neuron starting at this end. So this end here is what we define as the front of the neuron, because this is the origin point for action potential within the neuron, and we'll talk about why that is. These long tree branch-like structures that we see outside of the main body of the neuron are what we call dendrites, and the name is not surprising if you recognize the prefix dendro is the Latin word for trees or tree branches because of their relatively obvious tree-like structure. Now, dendrites' job is to receive action potential from something that came before it. Now, a lot of the time, there can be other neurons that are connected to these dendrites here, but if this neuron were a sensory neuron, such as a neuron that's connected to the human eye, this would be an example of where the action potential comes from a sensory organ like the eye. Now, the job of the dendrites is to collect action potential, again, either from a sensory organ such as the eye, or even multiple sensory organs such as taste receptors in the tongue or individual skin cells, and transfer that action potential down to the large central mass of the cell, which is what we call the cell body. So inside of the cell body are all of the main organelles, such as the nucleus, endoplasmic reticulum, mitochondria, Golgi complex, etc., and therefore all of the regular cell processes, such as protein synthesis, occur within the cell body. But in terms of the specific function related to action potential, the job of the cell body is to collect action potential from all of the dendrites, and assuming that the action potential that reaches the cell body is strong enough, meaning that there is enough action potential flowing from the multiple sources that this neuron is connected to, the neuron via the cell body then makes the decision if the stimulus is strong enough. The cell body is ultimately responsible for passing that action potential onto the next neuron, and the way that it does this is by taking all of the electrochemical action potential and transmitting it down this long canal here, which is what we call the axon. Now, eventually, we can see that the axon does not have a single endpoint, but actually multiple endpoints, and this is what we refer to as the axon terminus, although perhaps more correctly we would refer to this as axon termini, because each of these are multiple endings. Now here, depending on what this neuron is connected to, it might be passing the action potential onto another neuron, or possibly to some sort of effector organ, such as a muscle, or perhaps some kind of gland. So this means that neurons, unlike a large number of other cells, actually have a front end and a back end. Action potential always begins at the dendrites and always ends at the axon termini, and only ever flows in one direction. But this will become more evident in future videos. Now, at the beginning of the video, I said that neurons are one cell, sort of, and the sort of is because of these structures that we find along the surface of the neuron's axon. Now, not every neuron contains structures like this, but the more often a neuron is used, the more likely these structures, which are what we call myelin sheaths, form. Myelin is actually a mixture of a combination of lipids and proteins that surrounds the cell or the axon cell membrane, and the actual functions of these become rather obvious when we look at them in more detail. So we say that neurons are actually a single cell, but that is not entirely correct, because the myelin is actually formed by specific cells that live outside of the neuron itself, which are what we call Schwann cells. And Schwann cells live outside of the axon, and in order to form myelin, they basically take their cell membrane and wrap it around and around and around the surface of the axon like this, so that we have double, triple, quadruple, even quintuple layers of cell membrane that form on the outside of the axon, which are what we call myelin sheaths. And myelin sheaths have two primary functions. As mentioned before, the more often a neuron is used, the more likely it is to form myelin sheaths like this, because the function of myelin sheaths primarily is to function as insulation. Pardon the typo there where I wrote insulin. Now, any time where you have electrical current flowing through a location, there is the possibility that the electrical current can discharge and be released and potentially cause damage to the outside of the electrical wire. And in the same way that electrical wires are usually wrapped with some sort of insulation material like rubber, myelin sheaths fulfill the same function to make sure that action potential is trapped within the neuron where it should remain and where it should not be able to leave. Now, the other function of myelin is to actually speed up the rate at which action potential is transferred, and to do that, we need to look at the gaps in between the individual sheaths of myelin. So action potential cannot actually travel directly or continuously through a myelinated neuron. So electrical current, that being action potential, cannot move in a continuous progression like this, mainly because lipids, specifically the phospholipid that are found within a cell membrane, are nonpolar. And if they are nonpolar, that means that electrical charge is not going to be drawn to that specific location. So instead, the way that action potential travels is that it moves in a skipping motion from one section of the axon that isn't blocked by myelin to the next, to the next, to the next, to the next, until it reaches the axon terminus. And as it turns out, this skipping motion results in a higher electrical current than would be possible rather than if the action potential moved continuously. And these spaces that are found in between the individual myelin sheaths are what we call nodes of Ranvier, or if you prefer en français, nodes of Ranvier. Now, when we get to an axon terminus, we can see that the axon terminus of one neuron and the dendrites of the next neuron do not actually physically contact one another. There's actually a very deliberate space that is left between the two neurons, and this space, or rather the junction where one axon terminus meets the dendrite indirectly, is what we call a synapse. And synapses are very important in terms of regulating how well action potential is able to pass from one neuron to the next neuron, so much so that this is an entire topic in the nervous system unit. Now, the idea is because the axon and dendrites of two neurons do not directly touch each other, neurons actually require specific hormones in order to transmit the action potential from one neuron to the next neuron. And because these hormones are specific to function within the nervous system, we have a nervous system specialized name for neurological hormones, and they are what we call neurotransmitters, which again will take up a very important role when we focus on the structure of how synapses are structured and function. In the next video, we're going to use our knowledge of neuron structure in order to examine the simplest of neurological pathways that occur whenever an involuntary reflex kicks in, and these are what we call reflex arcs.